omics, and this is very basic slides, and each of you may have learned this, understood it. So when we call multi-omics, so let's dissect a cell. In the nucleus there is a DNA, and any modifications happening at the level of DNA, so we call genomic changes at the DNA, at the level of DNA, which could be genomic and epigenetics or epigenomics. Then there is a genome, so DNA becomes RNA. So we call genomic changes, then we call about the changes at the level of RNA. And this is what we call it transcriptomics. And then from RNA it becomes protein. And very basically you understand the protein, which is the basic unit of a cellular morphology or the phenotype. So genome, transcriptome and proteome. But now it is very interesting that when we talk about the protein, we also talk about the modifications at the level of protein, which call it post-translational modifications. It could be multiple kind of modifications, mainly phosphorylation we talk. Then metabolomics and cell signaling. So now what is very important, each of you look at this from left to right. When we go dissecting from DNA, RNA, protein, actually what we try to understand is from genotype to phenotype. We talk about the DNA, but what you express or either on the skin or your hair color, say keratin or melanin, so or dentine, all this is proteins we are talking. So what is very important that when we dissect genome, we should go from left to right, because what it is going to give you is the protein or the phenotype. And any such alterations at every level, what it will give you eventually, the cell signaling pathway. Each of the molecule, if you connect each of the molecule, you would like to understand that what has happened wrong in the cellular process. And it could happen for any such disease, not necessarily cancer only. So multi-omics. Now just go a little deep dive and understand that where the origin is started. When we talk about the genomics, I am talking about the era from 1950s to 1960s, where we have discovered, the first time we understood that there is a double helix DNA. And then we understood the genetic code, Sanger sequencing, where we are talking almost 1970s. I mean, still we are using Sanger sequencing in the lab. But then you go forward, we got the PCR developed, Human Genome Project. And this Human Genome Project, first human chromosome sequenced, Human Genome Project completed 80 somewhere. And then we just evolved. We come up with one after other technology. We talk about the MySeq, we talk about the HiSeq. And then the different kind of technology has developed. We'll talk about the NovaSeq, nanopore. So all this is a technology which actually leveraged us to sequence the genome very quick, fast, with the less price. Now, epigenetics, when we talk, we are talking quite at the age where 1960s, we understood that there is histone methylation. And then we move forward about the discovery of DNA methylation, discovery of histone ubiquitination, and bisulfite sequencing. So all this is coming around 1990s. So we evolved over the time. We understood genome, we dissected the genome. Then we came about the transcriptome, which was quite late in 90s. We understood about the microarray, and there was a time we just using the microarray. Now we talk about the whole of the transcriptomics. Proteome. So proteomics was pretty earlier when we understood MSMS, and then 2D gel electrophoresis, MALDI, and now we talk about all Orbitrap, first generation sequencing, second generation sequencing. And this is where we talk about the labeling and TMT labeling, 10x, 3x, 5x, and 18x. So we evolved with the technology with the time. Metabolomics again, lipidomics, and each of this component at the end, what it is giving us, the big data set. No matter what you deal with any such omics, you are going to get a big data set. And this big data set will give you eventually to go deep dive, understand, and make sense of it. So multi-omics for precision medicine. And very simple way, because you all are students sitting and you keep hearing now this term precision medicine. What does it mean? The name itself suggests precise. So how can you use any medicine which can effectively, precisely work? You get a headache, you go and get a drug, simple drug, and you may feel, some of may be feeling good, some may not respond. You go over the counter, you get a medicine for fever. And these are multiple medicine which you will get over the counter. Some of them are effective, some of them are not effective. But when you go and understand more complex diseases like cancer, we need to understand that what is the cause, what kind of drug should be given for that particular cohort. And this is where the precision medicine evolved. Now when we talk about the precision medicine, I talk about the omics data. And omics include every omics, multi-omics, where genome, transcriptome, proteome, methylome. But what it happens that we would like to curate the data from the multi-omics to identify the, or to subgroup the patients. And subgrouping of the patient means it could be ethically, environmentally, what is the origin, what you see, and this is where the patient got stratified because we evolved the targeted therapy. And I'll give you a beautiful example that what does it mean the targeted therapy. Now, what you see that the publications, if you go in the domain, you PubMed it, you will find that there are multiple data set has been developed, multiple publications. Genomics, you put